Good morning, everyone, and welcome to the Future Ventures podcast, Scaling with Clarity. Today's guest is Dr. Henry Erdley, co-founder of GoCART Therapeutics. Henry is working at the intersection of biology and engineering, focused on improving how we treat cancer using cell therapy. Before starting GoCART, he spent years in research looking at how immune cells can be programmed to better recognize and attack tumors. What he's building now is aimed at solving one of the biggest challenges in CAR T therapy: how to make these treatments more precise, more controllable, and ultimately safer for patients. This is a conversation about what it actually takes to build in biotech, where science, capital, and real world impact all collide. Welcome to the stage, Henry. I'm super excited to have you on and talk about CAR T therapy and your company. So, w- and my understanding is that you're visiting family now in Western Europe, in Western Germany. So, tell us a little bit about where you are, where you're calling from, and what's your
Yeah.
origin story.
Okay, so I'm currently visiting family over the Easter holidays. So I'm in a small town in Black Forest. It's called Freudenstadt. That translates into happy town. That's actually real. So, well, it got a lot of nature. So,
Yeah.
spending some time. But normally I am based in Berlin, where I'm an alumnus of the Charité Medical School and the Max Center for Molecular Medicine, which is a
Hmm.
Helmholtz Center.
Hmm.
And well, that's how I got into CAR T cells.
And why did you get into CAR T cells? Like, why did you decide to pursue the medical path? Like, tell me more as to what's driving you.
Also, I don't know, sometimes ideas kind of stick, you know. So, actually, the first time I heard of the existence of CAR T cell, which is a specific cancer immunotherapy where you genetically engineer cells to hunt down the tumors, and the origin that actually in high school, where I did a presentation on gene therapy in general, and then I read about, yeah, you can like get immune cells out of the patient, then you make them have an artificial receptor and with that they can hunt
Mhm.
down the tumors and kill the cells off. And that idea was kind of impressing, and it stuck in the back of my head. And later on, I went on to study medicine, and from beginning on, I always was thrilled about research and wanted to
Mhm.
do more in research and have a bigger doctoral thesis in there. And then I remembered, okay, yeah, that CAR T cell stuff that was so thrilling to you. Maybe you find and do something serious in that space.
Mhm.
I looked around.
And yeah.
So looked around and found the research group of Dr. Armin Ray at the Max Center for Molecular Medicine, a
Mhm.
sister institute that is also in Berlin University Alliance. I was studying at the Charité, which is the local medical school. And so I got in, and that actually started off with how to design a chimera's antigen receptor, how to tune it to a specific antigen. And I ended up with a project where I actually used an existing BCMA CAR on NK cells.
Mhm.
They have some disadvantages and advantages, and the main advantage of those is you can use them allogenically. That means you can take donor cells and give them to a patient, and in that case you can treat people with CAR NK cells even if their own T cells are destroyed by previous treatment, which is around
Mhm.
30% of patients, uh, and then fail to express CAR T cells, but you can use CAR NK cells for those cases. And I did a project to better tune them into the tumor niche, make them migrate to the tumor, and also
Mhm.
designed the first CAR that has multiple targets. That time it was a so-called OR gate, where it kills either target, whatever molecule you want you have on there.
Mm-hmm.
And that's how the problem got into my mind. So the OR gate is nice. It's a good backup mechanism should the tumor get resistant or lose one of these antigens. So an antigen
Mm-hmm.
is a protein on the surface of a cancer cell,
Mm-hmm.
and you want to train it down and kill the cell with it. But actually, there is a big problem. Most good cancer antigens cannot be used. We call them undruggable antigens.
Why?
And yes, that exactly is because, CAR T cells and any targeted treatment approach for that matter triggers all the cells that have this target.
Mm-hmm.
So you can only choose targets that are on the cancer, of course, and then on some tissue, some kind of cell, you could lose and still live somewhat comfortably.
Okay.
But for example, like CD56 and most good cancer antigens, highly expressed on tumors, can be downregulated, a driving factor necessary for proliferation, so the tumor cannot easily lose that, but is on the brain. Is on like every neuron, every nerve cell in your brain. And if you kill your cancer and
I see a
kill your
problem.
brain in the process,
Yeah.
that's kind of
Defeats the
counterproductive.
purpose. Yeah.
Yes, but it might be a successful treatment, but I don't think your patients will thank you a lot for doing that.
Yeah.
So, and that's actually kind of situation which is the normal case. So most good cancer antigens are in the nephron, on the brain, hugely expressed on bone marrow or some kind of essential tissue. And that
Okay.
actually leaves 70% of all cancer types out of a specific treatment and also out of CAR T cells. So yeah, that's basically the main problem we try to solve with GoCART, where we develop a system that recognizes two antigens again, but we can match them. We can match them, and our cells need both. We need both antigens to kill, and then we can combine them in a way that only the tumor has them. So we could use CD56 as said, and CD38. So one is on the brain, and the other one is on the bone marrow. Both things you'd rather not lose, but they're never together in nature.
Mm-hmm.
But they are together on the tumor, mainly on the acute myeloid leukemia and the multiple myeloma. So we started designing a CAR T cell system that can recognize this and selectively kill off the cells that have both, but not the cells that have only one.
Okay, so if only one of the antigen is present, then no good. If the two of them are present, then in this case, using the CAR T method, you're attacking just those cells because you know that those are cancerous cells.
Yes, exactly. We can choose
Okay.
these. We can choose these in a way that we know for sure that this is cancer and not
Okay.
your brain.
And then once you identify the cells, how do you attack the cancer?
That's actually the normal system. So we basically leverage the process your immune system uses for cancer cells and infected cells by standard.
Mm-hmm.
So basically what we do, we take T cells or NK cells. These are killer cells. They normally roam around your body and recognize unusual or altered cells that are that express something that looks strange, or that or for the T cell some specific pattern that that is not in your body.
Okay.
So, for example, a viral protein like the COVID S protein, or a mutated or dysregulated protein or stress proteins. And if they recognize that, they activate their intracellular killing mechanism. So a cascade happens that enhances the signal if these signals are on a cell the killer cell connects to. And then these cells have two steps. First, they try like knocking on that cell, if you want, like, so they have a receptor system like Fas and Fas ligand. So every cell in your body normally has Fas receptor, and the CAR T cell first tries to show Fas ligand to them. And if the cell somewhat works how it normally should, then this Fas ligand activates the cell's self-destruct switch, if you want. So the cell then starts to digest itself and die. If the cell is already beyond repair and the virus has completely reprogrammed it, or it is completely mutated into a cancer cell that has no reaction to Fas whatsoever, then the CAR T cell uses its second weapon, which is actually like they have vesicles, like bubbles of digestive proteins inside them, and if a cell does not respond to this suicide switch, then they throw out these digestive proteins onto that cell and actually destroy the cells on their own. So this is how the immune cells normally kills off your cancer cells and virus infected cells. And actually, that's the fascinating part about that. So if someone has cancer, that means this process must have already failed at some point. There must be some kind of mutated form of cell, the cancer cells, that escaped this recognition and were able to avoid your T and NK cells that normally kill off all the cancer cells you produce. We produce around 100 new cancer cells every day, and they get killed off. So if you have cancer, one of those will have gotten through the screening.
Okay.
And with CAR T cell therapy, we use an artificial receptor. We just put an artificial receptor on the cell that recognizes something on the tumor we choose to basically restore your immune cells' own healing power, to restore their ability to recognize and kill off this type of cell.
So is this like the least invasive way of treating cancer, essentially using your body's own immune system as the killer of the cells? Kind of like, how would you compare this to alternate cancer therapies?
Well, depends on how you define invasive. So in terms of change, okay, you genetically engineer a cell, but then it's your own cell. You could call it least invasive, but on the other hand, we must say this is a side effect heavy therapy, because high immune cell activation can actually create even life-threatening side effects. One example is so-called cytokine release syndrome, where a lot of tumor destruction and a lot of activation of killer cells floods your body with immune mediators, and you get like inflammation everywhere. So that actually the initial CAR T cell infusion always happens in a hospital and cancer center setting. So you cannot do that at home. But basically, once you get past the first part, then you, especially within T cells, have a long-term surveillance. So these cells form CAR T memory cells. They stay in your body and can find tumor cells that might come up later and still keep killing them. And that actually is an extremely effective kind of therapy. So when the first CAR T cells, CD19 CAR T cells came out, we see 1/3 of relapsed and refractory leukemia patients, which were on those study, cured. And these are patients that would be palliated otherwise. So there was no... We have exhausted all the chemotherapy and radiotherapy options. And normally we would say, okay, we treat this patient to the best supportive care we can, but they will ultimately die from their disease. And even in that situation, this kind of therapy could cure 1/3 of them,
Mm-hmm.
And another third of them was tumor-free for several years after that. So that actually is pretty strong results. So it's extremely powerful, and can change the course of the life of a person by quite a lot.
So, Henry, does this mean that from a treatment perspective that what are the avenues that CAR T therapies are applied to patients that are entering palliative care and all other alternate forms of treatments have failed? Or would you like to see CAR T as a method of treating patients earlier in their career cancer progression, as well as would you? I'm just trying to kind of understand from a market perspective, or would you focus on specific cancer types because this type of treatment would be the most effective?
Yes, so basically, a bit of all of that is the
Okay.
reality. So at the moment, we got approved ones for treatments of B-cell cancers. And on the one hand, myeloid cancers are easier to access for immune cells. So we see first results with adaptations of the cells. You need to engineer them more to be effective in solid tumors. But in blood cancers, we know it
Mm-hmm.
to work quite well. And so we are kind of moving from a niche into a broader application. And we with GoCART want to help and accelerate that by abolishing the undruggable antigen problem. So in general, we started off with hematologic tumors, and we at GoCART will do the same to prove the platform. But from
Yeah.
there on, relapsed and refractory hematologic tumors, it kind of moves out the ladder. So we already see subsequent clinical trials using it earlier in treatment and still showing great results. So I expect it to grow at least to a second line treatment within the next 2 decades. On the other hand, there is another issue that is raised by that. So the costs of this, at least as it's done at the moment, are rather high. That's also something GoCART tries to tap into by using in-house production methods. So we can actually have the cells produced at the hospital and just send like the hospital the tools to genetically engineer the cells, vector virus, for example, so that they do not have to send the cells to us, or fly them around the globe, fly them back, freeze and thaw them twice. So that actually is kind of a regulatory nightmare. So it is necessary at the moment for regulatory purposes, but actually each freeze thaw cycles makes your cell product a bit more shitty. If the patient already is severely pretreated with chemotherapy, the cells might be bad from the beginning on. I see these in-house production unit as a huge tool to, on the one, push down costs by quite a lot, and on the second one, improve quality of the cell product. And then pushing down costs, also what we see as a leverage is a modular approach. So like, for example, GoCART uses a common CAR T cell that is used for every indication now. So not one single CAR for every type of target antigen, but one for everything. And then we have a sandbox mode where you recombine specific binders. You give them subcutaneously. The patient gets an injection on a regular basis. And redirect the cells by this. So we can use the same vector, the same scaled cell product for all the indications, because that actually is the big cost driver, the cell product. So there are different leverages to push down costs from, at the moment it's 400 to 500K in Europe and up to $800K in the US.
Mm-hmm.
I think when we go to market, we could push it to 200K with our platform approach. And maybe in the long term, we can reach the lower double-digit thousands for one treatment, which would be, of course, the ideal outcome, and that would help scaling into earlier stages by quite a lot.
Yeah.
At the moment, the bottleneck is infrastructure for cell product production.
And so when you say getting into in the future, getting into double digits, like are we talking like 10,000, 20,000? How many treatment would a patient need? What do you think it's possible or achievable? What would be a huge unlock on both? I mean, on the quality side, I get it. From a cost and velocity perspective, what would be the huge unlocks for you, or this type of therapy to get it down into really approachable territories where we're talking like single-digit thousands of dollars for a per treatment?
A single-digit thousand, I think that's a bit unrealistic.
Okay.
Because at the moment, because in the end you have to extract the cells from a patient. Then you have to treat them, even though semi-automatic units become available. You need the relevant staff to do that. And even without shipping, it takes like 1 to 2 weeks to do that. You need to you need to culture, transduce, and expand the cells to reach a relevant amount of cells at suitable engineering levels, like we call the transduction rates. How much of this cell product is actually successfully genetically engineered, and how much not? And you of course want something ideally 40% or above before you want to give it back to the patient, and that's actually something that requires work and specialized staff in the end. But what we currently have is like estimates of cost of goods, like the pure production costs of around $60,000 per one cell product for one patient.
Okay.
And as said, by standardizing this and having like the same vector for all the indications out there, is something like we can use with a modular approach. We can use economics of scale and push it down to maybe the lower double-digit thousands.
Okay.
And then these binders are there to redirect the cells over time. So this can make it more accessible in the long term.
How would you use AI to drive velocity and drive cost?
Yeah, actually, we use AI for development of an interface. So what we at GoCART actually do is we have these AND gate, these double recognition, and it works with our binders. So what we have is we have 2 different binders you choose. For example, CD56 and CD38 a set for AML and multiple myeloma. And that swims around in your body and attaches to its targets on the tumor cell. And if it's a tumor cell that has both of them, then they can heterodimerize. So we have a peptide engineered that kind of sticks together. So the 2 different of those can stick together, and our CAR T cell is engineered to only stick to these heterodimers. So only if they're stuck together, our cell sticks to that and gets activated. And if there's only one on a cell, then our CAR T cell does not stick to the single one. It needs both of these to stick together. And that actually is what is enabled by ML-driven protein engineering. So we with the novel methods of binding prediction, we designed peptides that actually can do this with a high fidelity, and even obtained these artificial candidates in the first place. So something that with a high fidelity will stick together, like A and B, I call them, which are the 2 difference on the binders on the tumor cell, and then C is on the CAR, on the killer cell.
Mm-hmm.
And we need to design them in a way that first A and B stick together, and only then C comes. And that's done by ML-driven binding
Mm-hmm.
prediction. So what we have is an engine that automatically creates and mutates sequences of this specific
Mm-hmm.
peptide type, and then tests their affinities against each other, and
Mm-hmm.
then spews out sequences that do what we want them to do. So at the moment, we have screened around 180,000 individual trimeric sequences, and out of this, there is a lot of noise produced and like semi-useful stuff that, well, can be used to have the AI iterate on it, but could not be a candidate.
Yeah.
And from these 180,000, actually, we identified 20 sequences that actually do what they should. And without AI-driven peptide engineering, that would just not be possible. Even the biggest pharma player could not produce all these different sequences for such a moonshot and test them in vitro. The costs would be astronomical. So testing one of those in vitro is around 1,200 euros.
Mm-hmm. Mm-hmm. Times
And
180. Yeah. Got it.
times 180,000,
Yeah.
that would probably get even big players in trouble. So
Got it.
these late advances actually are what makes this possible in the first place.
Mm-hmm.
We can select these prime candidates, and now we test these 20 candidates.
Yes.
And that's rather doable.
So, I mean, obviously, AI has been a huge unlock for you in terms of driving precision and quality. Do you believe that cancer will be eradicated as a disease in the future?
What is eradicated? So I think we can treat it. We can treat it more. Actually, cancer is kind of like a hiccup of your natural process. So it's not like an infectious disease you could find a
Yeah.
vector for and then eradicate that like
I
for
mean,
smallpox.
you,
But what I
you
think
mentioned that
is,
your body produces 100 cancerous cells a day.
yes,
Are you able to essentially kind of like you're German, and Germany
yeah,
is known for having some of the best performing automobiles. Can you get the engine tuned up in such a way that it just it's it's performing in such a way that like there's never cancerous like that antigens like
never
that.
surface in the first place, so you
Yeah,
never.
exactly.
Yeah, that's kind of an interesting take, like having having pre-built CAR T cells in your body, or having your immune cells tuned up to the max. But actually, that's kind of an interesting thing to think about. But you have to know the dirty truth about the immune system. The immune system can cause horrible diseases by being hyper aggressive itself.
Okay.
That's
Okay.
the whole field of autoimmunity. And actually, evolution put the immune system where it is, in kind of a sweet spot between not allowing too much cancer, but also not getting everyone a horrible autoimmune disease.
Yeah.
So actually, kind of like engineering that, meddling with that, will probably need a lot of care if you try that.
Understood. So there's a fine balance.
Yeah. So actually, it's pretty fine balance. And what I see as the future is in cancer, well, we see that in this case, the balance kind of failed on the cancer side, and a mutated cell type, what we call cancer, got through.
Mm-hmm.
And then we can use our genetic tools like the
Yeah.
CAR T cells to hunt that down. And with systems like ours, we have a lot of flexibility. We can in the ideal future tune it down to exactly what this tumor is. So
Got it.
as we have this binder approach, these adapters, we start with these two binders and approve it in AML and MM to get on the market. To have like a clear path to market and an orphan designation, orphan approval, which saves a lot of money, which should normally is used on a phase 3. We can use the orphan designation to get around this, or at least already have a sellable product when doing the phase 3 follow-up. And from there on, we want to expand. So we are open to any collaborators. We already have 3 of those who develop antibodies, VH8 binders in the broadest sense. They need to be a single chain peptide binder. And they can step on our system. You can hang on our interface that we develop for low commission, like 5% or something. And then they can start approve and sell off these binders that fit our system, or we buy the patent of them and further develop it at some point. And then we deliver the CAR T cells, which are kind of like the operating system. And the treating physician of a cancer patient can phenotype this tumor. You can take histology out of this tumor and really find out what is on this specific tumor of this specific patient. Not only the statistic average, but this patient. And then you can recombine these binders and also backup binders if you need them for like tumors where half of the tumor has one antigen and the other half another one. Tumor cells constantly mutate and change. And then you can use our CAR T cells, these binders, and have a system that backs that up and kills them off. And also modularity can be used to change the course of this treatment if needed. So should one of the antigens be lost, the tumor stops to express one of them, loses one of the targets. Normally the CAR T cells would fail at that point, but with us you could look into the expression again, look for a new target, and then change to that new binder, and it should keep working. So
interesting. So if I would translate it into my own words for somebody that doesn't have a PhD or doesn't have the medical biotechnology background, essentially we're going from one-to-many medicine, we're driving precision medicine, we're going to one-to-one. We start with a phenotype analysis of the particular patient in mind, and based on this, we're reconstructing what kind of binders we attach to this so that it's unique and precise to the patient. And depending on if it's work or it doesn't work, we kind of fine-tune it. We'd add backup binders, so we add different binders that help to attack the understood.
Yes, that's actually the vision. So approaches like this are called personalized medicine. We always had the discussion how to make personalized medicines, because when the term came up, it was like designing a whole new therapeutic for one specific patient, and that's extremely expensive. That's extremely expensive and not really feasible. So that's like kind of an idea to have it modularized, to have it recombinable, to deliver the huge advantages, and that have been shown clinically, of a personalized approach, while still having all the components as an industrial upscaled product that can be delivered at sustainable costs for our healthcare system.
So in essence, is the vision for GoCART to become the platform that builds out, to use your words, to build out the operating system, and all of these different partners that you have are just developing binders and binders and binders, and then you continue to provide that personalization and precision, and then you have the partners that are kind of the ecosystem around you.
Yes, so basically we want to be maximum open with the platform, because that of course also enhances the value of the platform from a company perspective as well as from a clinical perspective, because the more binders out there, the more different antigens on the market, the more options for combination, the more backups a physician can give his patient with that specific treatment approach. And therefore we want to design an environment where we provide like the CAR T cells, the operating system that delivers the end gate with these binders, where you can use these two different antigens to kill a cancer cell. So we can actually use the undruggable antigens that at the moment you can use them for diagnosis, but not for treatment. With this you can. And
Okay.
everyone out there, so in theory, so first antibody development companies, of course, because we can collaborate on a project base with them, and they can approve their own binders. But at some point, we could also we want to also expand to R&D teams at universities or something. So also in the long term, the vision is that we offer collaboration like for low commission, where actually the partner does the validation and then the clinical trial for a specific binder. Or at some point we would like buy off binders from academic labs, or we work together with academic labs to further develop it then in-house once they found an interesting binder for a platform and we want to add it. So the more binders on the platform, the more powerful the treatment is.
Valuable the platform.
Yes.
And so, speaking of treatments, if somebody if we have a patient or a loved one of a patient, and somebody's hearing that the podcast, how and when can they get this type of treatment?
So basically, at the moment, well, GoCART, of course, is at the very early stage. We are moving from in silico to in vitro, so we would need until 2033 to get the orphan to proceed to the orphan approval. But you might want to look into other specific treatment approaches. Talk to your oncologist. There are already approved CAR T regimens if you got like multiple myeloma, diffuse large cell B cell lymphoma, or ALL, acute lymphoblastic leukemia. Then there are approved regimens out there. Then an inclined oncologist should always look for clinicaltrials.gov. Because actually, in many situations where standard treatment fails, and in some cases we already have very good standard treatment. So, CHOP for lymphoproliferative malignancies is actually rather powerful and has been developed over the last a bit more than a decade.
Yeah.
And sometimes it makes sense to enroll in a clinical trial with something that's already being developed and is being tested against standard treatment. So that's something you need to know about clinical trial. You always get at least the equivalent to the standard of care where it's tested against, and and potentially you're getting something better. Of course, you need to be aware that this is then like not completely validated, dependent on the stage you are stepping in. Is it a phase 1? Then even toxicity studies still have to happen. So that's also like a risk you take. But this actually generates data for us for us as researchers to move these types of novel treatment into the validated space and onto the large public market, and also can potentially deliver better results for you as a patient or a loved one.
So Henry, you said that the data is a key component. What has been the biggest technical challenge that you have solved today, and what's the one standing in the way going forward?
Yeah, the biggest technical challenge was to automate these binder prediction.
Okay.
So basically, 180,000 sequences, designing them by hand and then running them through a binding predictor is not so feasible.
Yeah.
So we need to automate that. So what we have at the moment is we have this iterative approach that originally used a homo-trimer template, so something that just sticks to itself, and then ran it through cycles and cycles, and it was mutated and then tested against each other and mutated again. And if it moves towards our metrics, then that one is used, as a template and mutate it further and further. So building this engine actually is the thing that created our valuable candidates in the end.
Yes.
And well, big ones moving forward. The next one is, of course, closing our angel round. And
How much are you looking to raise?
150K.
Okay.
So that's possible through our collaboration with the University of Cadiz, where actually configurations of CAR T cells plus binders are already being developed and tested. So we can use pretty strong synergies. Rather happy to have this collaboration. We could decrease the ask from 500K to 150K through that. And we are currently starting in vitro validation. So we express these binders in a human cell line.
Yeah.
And we want to find the binders that actually do the same as predicted in silico. And from then on, we express them as a CAR. And then we do my favorite experiment, which I've been done in the whole time I've been in CAR NK and CAR T cells, the co-culture. You take a tumor cell line, throw it into a bucket, if you want so, and then throw in your effector, your killer cell. And with the binders, we of course throw in the binders in the configurations we want, and then we want to see
the effect.
Tumor cells plus both binders and both antigens killed. Tumor cells with only one binder or one antigen, they should not be killed, because that would then be your brain.
Yep.
And that's the next exciting path, validation parts. So from then on, we can move into NSG mouse validation, which is like a mouse that has no immune system, but and that means you can test human cells in them. So that's kind of like 1 year in vitro in cell culture, and then 2 years in the mouse, and then we could move into the first clinical trial, or at least file for moving into IND enabling, and starting a clinical trial with the first actual patients.
Understood. And from our prior conversation, you said you have a great relationship with the University of Cadiz, and one of the leading researchers, I don't remember the name of the professor, is collaborating with you on the development.
Yes, so it's the Institute of Francisco Garcia Collar. He's department leader at INIBICA Cadiz, so the Institute for Biomedical and Biological Research down there. And they actually developed like CAR plus one binder system, so where you could already have a CAR and then swap out different binders for different targets, like what I said. If a tumor gets resistant, you could swap out, or you could use 2 different binders, and then the CAR T cell will activate on both targets. And we actually fit in neatly with that. So we have our double binders. So we add the AND gate onto this binder approach, where we have like 2 different binders that must be together, and that's the new one. And we can use all the infrastructure down there. So we have the experiments established. There's actually only one that is new. That's the expression in human cell line, because that's necessary for our specific type of interface peptide. But all the other validation experiments are already in there, material already in there, machinery already in there. We can use the laboratory. And it's basically a very startup-friendly university. So we have a sponsored research agreement in place. Questions around patents, present or future, are already solved, contractually even. So that's.
And do they remain with you in terms of any kind of intellectual property that's developed?
Yes, that's actually what it says. So intellectual property from this project will remain with GoCART Therapeutics. And we provide 125K in 2 tranches of research proceeds. So that's basically kind of like the use of this 150K. 25K stays within the company for contingency. And in these 2 tranches, first 75K at the beginning for the binder validation phase, like the inhuman cell line expression and this stuff. and the second tranche when we move to the
Hello?
I think we lost each other for a short time. What was
Yeah.
the last I said?
I don't remember, but we'll have to edit this part. So in terms of the research, the angel round 150K, it primarily goes towards the University of Curtis for furthering the research. How much of a runway is this giving you, Henry? Like, are we like 6 months, a year, kind of like, and obviously we need to find a way to continue to fund you to be able to scale the research. So kind of give me a sense on the capital side, kind of what are the
So,
requirements?
yeah, basically that is even a 1 year runway. So
Okay.
it funds a 12-year project, and
Okay.
that is possible because much of material and machinery is already there, and we do not
Yeah.
have to pay for the yeah, for the for lab rent or something like that. So that's like already in the package, and we need and is for research proceeds, material, and salary. So it's it's a 1 year package, and the milestones are validation of the trimer, and that's
Mm-hmm.
the big patenting milestone. So
Mm-hmm.
that might even happen within weeks that we file the patent, the core patent for the whole system
Mm-hmm.
with this data that comes from that. And the second milestone is validation of cell killing in this co-culture experiment, and that actually is the major milestone. So like the in vitro phase is covered by this,
Mm-hmm.
and then the next race, that is a 5M race, probably a bit less if several grant applications that we filed get through.
Yes.
That will fund the mouse phase. Also, perk is the university already has a running mouse trial approval for CAR plus adapter configurations. So
Mm-hmm.
saves a lot of bureaucracy to just expand this instead of filing a new one.
Mm-hmm. Mm-hmm.
And yeah, actually, that's also what or already interested investment funds in the second round told us. So basically, this angel round funds us to the milestone where we already have soft commitment.
Got it. Got it. Okay.
And yeah,
I mean that's pretty exciting. Now I know we're coming on to time. If somebody wants to pull out a checkbook and write a check, if somebody wants to follow your work, if they want to get in touch with you, what's the best way to do so?
we have a website, gocarttherapeutics.com, where you have like a rundown on a project and on a tech.
Mhm.
You could use the info@gocarttherapeutics.com to write a mail to us.
Okay.
We are on LinkedIn. I personally, Henry Erdley, am on LinkedIn. Or you could reach out by henry.erdley@charité.de,
Okay. Okay.
which is
Fantastic.
which is also possible.
We'll drop those in the show notes. That's fantastic. Kind of like a question that is a curiosity question for me. What's a belief you hold about the future of medicine that most people would disagree with? Kind of like, where do you see future of medicine moving into?
I think genetic engineering will play a core role in it.
Okay,
Two things will happen. Well, or I think at least they
already
should
happening.
happen
Okay,
if we
okay,
can get regulators to go with them. That is one, that treatments are even more engineered to a specific patient needs.
yes,
We modify cell types in vitro or ex vivo, like the CAR T cells, or in vivo, like for some orphan drugs that have
hmm,
gene therapies. And and it will get more modular, more patient individual, and actually it will move away a bit from like the current standard of completely validated clinical trials. So we will, for example, with us in GoCART, there will be clinical trials, of course, validating each binders for one disease. But I think doctors with this will get more freedom to recombine and use different, maybe more patient individual approaches. And that's also something I hope regulators will endorse, because we already have clinical results that show that these individualized attempts have higher success rate, higher sustained complete remission rates than it might be the fully approved and guideline correct treatment line. We see first steps in the right direction in what I say is the FDA Modernization Act that has been started in the US and is currently being put into actual law. So with the with a pathway-based approach for compassionate use that actually gives individual physician more freedom to try and use patient individualized recombinations and treatment approaches. So that's actually something where I think the future lies. But to get to that future, we need regulators to move with us. We can help that with data. And on the other hand, of course, it will get more modular, because the modular approach is necessary to deliver this at somewhat sustainable prices. Also, another thing, if we want to look into drug prices, we might think that what today is called the orphan drug, the orphan pathway for like situations like relapsed refractory AML, where the patient would have to be palliated with only best supportive care, where it is allowed by FDA and EMA to get on the market after a phase 2 if you can prove substantial added value in these orphan situations. And I think we should have an honest societal discussion about making this not the orphan pathway, but the standard pathway. Because already with the orphan pathway, we are at probably in most cases triple-digit million tickets for like the whole party to approve a new medicine, and the phase 3 alone adds another triple-digit million. And what it delivers is data on how long does the treatment work and what are rare side effects, and that takes years. So I think we might want to balance more between added value to a patient, because after phase 2 we at least know it works.
Yes.
And then we might have it approved, allow developers to earn some money and not have to exorbitantly inflate prices to break even after spending another triple-digit million.
Yeah, makes sense.
And at the same time, give the decision to the patients. Of course, they need, and this is where the physicians come in. We have to make an informed decision. We call it the informed consent to choose a novel therapy approach, or to stick with the standard of care and being on the market after phase 2. I think can bring down the costs in our healthcare systems by quite a lot, because this is the validation costs, not even the development and design itself, but the validation, safety, and then the clinical trials that are the main cost drivers for novel and patented therapies.
Makes sense.
Yeah.
So it's almost like running a live trial after phase 2, where you're continuing the journey with a patient.
Yeah, that's basically the same in the orphan orphan approval pathway. This does not mean you you scrap the phase 3 and do not do reports and forget about potential side effects. No, you still do all that research, but you do it while
Mhm.
on the market, and
Yeah.
that allows you for much larger groups.
Yeah.
So much more patients will report data. That's actually we call that phase 4. That's like market
Yeah.
surveillance. Once we are on the market, we observe the patients long term and add
Mhm.
that to the database on that specific treatment. Has to go on for 10 years.
Mhm.
And you still do this with the today orphan approach. So
Mhm.
in the end, the data might be even more valuable, and
Mhm.
you are able to deliver value to a larger number of patients at
Yeah.
an earlier stage, and in the end get a new therapy out there at
Make-
lower costs, which in the end should be the interest of us all.
Well, I mean, if anything, the speed, the efficiency has a lot of benefits that it brings along with it. Which one of the big unlocks is more data means higher quality, more insights. But yes, it's not necessarily without risks, but the risks could be somewhat mitigated through informed consent. And so if you have this effective monitoring mechanism, you can you can adjust, you can pivot along the way.
Yes, indeed.
Okay. Henry, it's been fantastic to have you on. I know it's Easter weekend, so go spend some time with the family.
Yes,
And
great pleasure to me as well, and
yeah,
wish you and all the audience happy Easter.
thank you. Thank you. Same to you. Okay.